What's up, Tumna Lectures? In this video, we are going to be looking at what we call the chemical digestion and absorption of what we call the proteins. If you find this video helpful, please make sure you like the video, comment down in the comment section, and then please subscribe. Let's go ahead and get started. In the previous video, we looked at what we call the basics in what we call the proteins. We looked at what we call the bonds, which we are going to be chemically breaking down. If you have noticed watching on that video, please make sure you go and watch the video so that you are now well acquainted with today's video, which is going to be mainly focusing on the detail of what we call chemical digestion and absorption of what we call proteins. We said that we are going to be breaking one of the bonds, which is going to be linking together the amino acids, which is called the peptide bonds. And we said that by breaking this peptide bond, we use that chemical process which was involving addition of water, which we called the hydrolysis. Okay, so now in this video, we are now going to be going into the detail and see how we're going to be breaking that bond chemically in the different parts within what we call the alimentary canal. Let's go ahead and get started. Now, before we go into the detail, we want to take some little bit of the anatomy and see how we call all these different areas, such that you also come aware of what's going to be happening in different areas within the alimentary canal. Now, this small portion here, this is going to be the portion of what we call the mouth. I have drawn it small because you're not going to be having chemical digestion of proteins within the oral cavity. Okay, it's all that simple. Now, this big dilated structure here, this one is representing the portion of the alimentary canal which called the stomach. We are going to be having the first kind of the chemical digestion of proteins starting in what we call the stomach. It's all that simple. Okay. And then this dilated structure here, this one here is going to be a portion of what we call the small intestine, the duodenum, the jejunum, and then the ileum. We are going to be having the final chemical digestion of the proteins and even eventually what we call the absorption. And then also have this cell, this one what we call the absorptive cell. This cell here is what we call the enterocyte. Okay, this is the cell which is going to be absorbing the final simplest protein molecules, which we call the amino acids. Okay. Then, of course, you're going to be having different kinds of the tubes that are going to be giving us different kinds of the secretions, which are going to be aiding in what we call the chemical digestion of the proteins. Okay, so now having the idea of the anatomy of this structure we are calling the alimentary canal, let's go ahead and get started. Here we have a big piece of what we call the fish. It's very delicious, nice to enjoy. But we want to first understand what is that kind of the macromolecule we are going to be getting from this kind of the big piece of the fish. And of course, we are going to be getting a lot of different what we call the proteins. Now, we are going to be having this long chain of what we call the protein molecules, which we call the polypeptide. A long chain of what we call the polypeptide, which is going to be having a lot of different kind of amino acids, which are going to be linked together by what we call the peptide bond. It's all that simple. You're going to be having a lot of different amino acids, and of course, we said that the body has approximately 20 different amino acids that are going to be differentiating from one another by what we call the R variable side chain. We talked about all that in the basics of what we call the proteins. Okay. Now, the breakdown of this kind of the long chain of the polypeptide is going to be a catabolic process. It's going to be a catabolic pathway, which is going to be leading into the final absorptive molecules, which we call the monomers, which are what we call the amino acids in this case. Okay. So now let's go ahead and get started and see how we are going to be breaking down this kind of what we call the peptide bond. Now, this kind of the long chain of what you call the polypeptide is going to be having the amino end, and it's also going to be having what you call the carboxyl end. That is the basic. Okay, it's the basic to know that. Now, within the stomach, this is where we're going to be having the first chemical digestion of what we call the proteins. Now, what's going to be happening is that when the food reaches into the stomach, we are going to be having that kind of the stretch, okay? That kind of the stretch. If you remember very well during the gastric phase, we said that when food reaches into the stomach, there's that kind of the stretch. Now, we have what we call stretch receptors that are coupled with sensory nerve fibers of what we call the vagus nerve. What happens is that our vagus nerve sensory fibers will take the information to the central nervous system, primarily to what we call the medulla oblongata. What happens is that within the medulla oblongata, we have what we call a nucleus called dorsal nucleus vagus, which will activate what we call the motor fibers or the vagus nerve, which will come out through the jugular filament and comes on what we call different cells within the structure we are holding in the gastric gland. what happens that vagus nerve is going to be releasing acetylcholine to M3 receptors on this super very important of this cell. You know how this cell is called? This cell here is what we call the chief cell. What happens is that under the influence of acetylcholine, this chief cell can release one of the most important molecule, which we call pepsinogen, which we call pepsinogen. Okay. Now this pepsinogen is in its inactive proteolytic enzyme. We need to activate it so that it becomes active to start the process of chemical digestion of what we call the peptide bonds. Again, vagus nerve is going to be releasing acetylcholine on this another kind of the cell, which we call the parietal cell. What happens that this parietal cell will release a molecule called hydrochloric acid. Now, it is this hydrochloric acid that maintains the pH of the stomach acidic. That kind of the acidity of the stomach is very important in activating this molecule calling pepsinogen into an active proteolytic enzyme, which we call pepsin. So now what happens is that this kind of the pepsinogen is going to get converted into another active form, which we are calling the pepsin. Okay, which we are calling the pepsin. So now, who was driving this factor? It was the hydrochloric acid, which was coming from what we call the parietal cell. Okay, so now generally we said the pH between 1.5 to around 3.8 is enough to convert what we call pepsinogen into an active pepsin, which is going to start the process of chemical digestion by breaking down different kind of what we call the bonds. Okay. Now, besides that, you are seeing that this kind of the coiled structure of the polypeptide, this is going to be like a primary structure of the protein. Okay. Now, what happens is that this kind of the primary structure of the protein, under the influence of hydrochloric acid also does another kind of the beautiful mechanism of making sure that it uncoils this long kind of chain of the polypeptide into this kind of unfolded chain of what we call the polypeptide. So now you can see that this coil under the influence of hydrochloric acid. Okay, under the influence of hydrochloric acid, it is trying to unfold this coiled structure of what we call the polypeptide. Now, this kind of the process is what we call denaturization of protein. Okay, this one here is what we call denaturization. Okay, this is what we call denaturization. Now, denaturization is the process by which what we call the primary structure of the protein is now unfolded. And this one is very, very important in a way that it is trying to expose the peptide bond to make it easy to be cut by what we call the enzyme pepsin. Okay, it is such another beautiful thing. So now, having seen that, now let's go ahead and see how is this enzyme we are calling the pepsin, how is it going to be coming and start cutting off this long chain of this kind of the polypeptide. Now, what happens is that this kind of the pepsin, once it has been activated, pepsin is very beautiful in a way that more of the pepsinogen, which are going to be coming from what we call the chief cells, they are going to be now getting activated by the already activated pepsin, meaning that pepsin is like auto-activator. It helps to activate the other kind of the pepsinogen, which has been activated by which has been released by what we call the chief cells. It is such a beautiful thing. Okay, so pepsin is also an auto-activator. Okay, now this pepsin, he likes to target those kind of the peptide bonds which are going to be on what we call the carboxyl end of this long chain of what we call the polypeptide, mainly targeting on the different types of what we call the amino acids. Now, this long chain of the polypeptide is going to be having a lot of different amino acids, but this kind of the pepsin is going to be targeting mainly those kind of the amino acids like phenylalanine, the tryptophan, and also what we call the tyrosine. It's going to be targeting those kind of the peptide bond that are going to be on facing what we call the carboxyl end of what we call this long chain of the amino acids. That's all that's going to be doing. Okay. Now, what happens is that if this kind of the pepsin comes over and breaks this bond here, it comes off on phenylalanine, it comes on tryptophan, it breaks that bond which is facing the carboxyl end, and also comes on the tyrosine and also breaks that kind of the bond which is facing what we call the carboxyl end. That's what it does mainly. Okay, so now what happens is that under the influence of the pepsin, we are going to be making a lot and a lot of different kind of the small kind of the fragments. What are these kind of these small kind of the fragments? They are going to be what we call small. They are going to be what we call small polypeptide chains. Okay, small polypeptide chains. So now we're going to be making a lot and a lot and a lot of the small polypeptide chains, which may even call what we call the peptones. Now, what happens that the peptones, they have specific type of cells that are sensitive to high levels of the what we call peptones. Within the antrum, we have this cell which we call the antro-G cell. This cell is sensitive to what we call partially digested proteins, which we call the peptones. What happens that this cell is going to be getting activated by peptone and releases a molecule called the gastrin, of which gastrin can move into the blood and comes over and stimulates the chief cells and the parietal cell to produce their own secretions. It stimulates the chief cell to produce pepsinogen, and it stimulates the parietal cell to produce hydrochloric acid. When you have these two molecules, pepsinogen will get converted to pepsin, which will continue breaking down this long chain of what we call the peptide. Such a beautiful thing, okay? So now pepsin is such a beautiful thing, targeting amino acids on what we call the phenylalanine, tryptophan, and what we call the tyrosine. Okay, so now we are forming these long, small polypeptide chains, a lot of them. Okay, so now that's what's going to be generally happening within the stomach. Now, moving ahead these things into the small portion of what we call the small intestine. Now, after that kind of the chemical digestion, we need to push these things into the small intestine. And of course, within the small intestine, primarily we're going to be having now the first portion of the small intestine, which we call the duodenum. Now, within the duodenum, we have this beautiful cell. This cell is super sensitive to what we call partially digested proteins, which we call the peptone. What happens is that this cell is what we call the eye cell. Okay, this cell is what we call the eye cell. This cell is going to get activated and it will release a molecule called cholecystokinin. Cholecystokinin is a hormone. It will move into the blood and it goes to a specific type of this structure, and this structure is what we call the pancreas. Within the pancreas, we have this bulbar structure, and this structure here is what we call the pancreatic acinar cell. What happens is that this structure will be stimulated by what we call cholecystokinin to release a lot of different kind of the enzymes. However, the enzymes of interest in this case are going to be the pancreatic proteases. Okay, we have a lot of pancreatic proteases that are going to be released by what we call the pancreas. They will come over and pass through this the sphincter, which we call the sphincter of Oddi. What happens that this sphincter of Oddi has receptors for cholecystokinin. It will come over and bind on those receptors and to make the sphincter of Oddi to relax and allow the pancreatic juice having a lot of pancreatic proteases to enter into what we call the duodenum. Now, the following are the examples of pancreatic proteases that you're going to be having. Okay. The first one is going to be what we call trypsinogen. What we call trypsinogen. Okay. And then the second one is what we call chymotrypsinogen. The third one is what we call carboxypeptidases. Okay, and then the last one is what we call the proelastases. Okay, the proelastases. Okay, so now when you see these are the four main kind of the protease enzymes that are going to be now continuing in the breakdown of different kinds of the small chains of what we call the polypeptide. Now, what happens is that all these these proteases enzymes here, they are in its inactive form. Okay, they are inactive. Okay, they are inactive. Now we need to activate these enzymes such that they can start now the process of cutting different types of the bonds. Okay, now what happens is that within the epithelial linings of what we call the small intestine, we have a super kind of this enzyme, and this enzyme is what we call enterokinase. You see this this enzyme here. This enzyme here is what we call enterokinase. Okay, this enzyme called enterokinase, he comes over, and then when trypsin comes over on this enzyme, he gets activated into an active form which we call the trypsin. Okay, so now what happens is that when trypsinogen comes over on this enzyme, it gets activated to another active form of the enzyme which we are calling the trypsin. Okay, Now, what happened is that trypsin is such a beautiful thing. What happened is that trypsin is also what we call auto-activator like pepsin. It can even come over and also activate the remaining kind of the trypsinogen back into what we call trypsin. It is such a beautiful thing, okay? So now, trypsin can come over and then activate this kind of other kind of trypsinogen into trypsin. So we shall have more and more of the activated kind of trypsin. Now, what's going to be happening here is that trypsin is a very, very kind of the friendly kind of the enzyme. You know what he does? He can come over and activate the rest of the kind of this kind of the protease enzymes. What is he going to do exactly? He will come over on this kind of the enzyme we are calling the chymotrypsin into what we call chymo chymotrypsin. Okay, it comes over and converts this enzyme which you are calling chymotrypsinogen, which is in inactive form, into what we call chymotrypsin, which is becoming now active. So who was driving this process? Trypsin is driving this process. Okay, it comes over and also activates what we call the pro-carboxypeptidases into what we call carboxypeptidases. Okay, so now trypsin will come over and also activate this kind of the enzyme into what we call carboxypeptidases. Okay, so who is driving this process? Again, trypsin is also doing a very important role here. Okay, and then the last one, which we call proelastases, again trypsin will come over and also activate this enzyme we are calling proelastases into an active form, which we are calling elastases. Okay, which we are calling elastasis. So now we have converted this enzyme from proelastasis, which is inactive, into the active form, which we call elastasis. Okay, which we call elastasis. So now, who was driving this process? Again, trypsin is also doing a beautiful work to convert inactive proelastasis into elastasis. Such a beautiful. So now trypsin is such a beautiful thing that can be activating all these kind of these proteases into their active form. Okay. Now, what is something which is very beautiful is that we are going to be seeing what is trypsin going to be doing, and what is chymotrypsin, what is going to be doing, and what are these kind of the carboxy and elastases, what are they going to be doing exactly. Okay. Now, we're going to be starting with what we call trypsin. Now, this kind of the enzyme which you are calling trypsin, trypsin, he comes over on this small kind of the small polypeptides, and he cuts what we call the peptide bond on mainly specific type of amino acids. He comes over on specific type of amino acids, mainly on what we call the arginine, and also what we call the okay. So now by doing that, it's going to be cutting off these bonds, So now by doing that, trypsin is going to be leading to the formation of the following kind of the molecules. He will lead into the formation of what we call the dipeptide, and even also may lead into the formation of what we call the tripeptide. Okay, so now trypsin is going to be leading to the formation of dipeptide and even also what we call the tripeptide. It's all that simple. Okay, now coming to what we call chymotrypsin. Now, when we come to chymotrypsin, what we call chymotrypsin. Chymotrypsin is a beautiful thing in the way that he cuts the same kind of the bonds which are on amino acids, like the same for what we call pepsin. Okay, so now we said that pepsin was targeting phenylalanine, it was targeting tryptophan, and also what we call tyrosine. Chymotrypsin, he does the same thing. He comes over and he cuts what we call on what we call the phenylalanine, and even also what we call the tyrosine, and even also on what we call the tryptophan. And also, this kind of chymotrypsin is also going to be leading into the formation of the following kind of the molecule. He will lead into the formation of what we call the dipeptide, and even also what we call the tripeptide. Such a beautiful thing. Okay. Now, another thing is that I also have what we call the carboxypeptidase and what we call the elastases. These enzymes, for them, they do almost the same thing. Okay, so now we have what we call carboxypeptidases and what we call elastases. Okay, so now what happens is that these two enzymes, they are super very important. in a way that they are going to be cutting off amino acids, which are going to be mainly on what we call the carboxyl end, okay, which are going to be on the carboxyl end of what we call this small polypeptide chains. What happens is that these carboxypeptidase and elastases will come over and break this bond, starting on what we call the carboxyl end. He comes over and breaks that bond. So now, by doing that, these two enzymes are going to be forming some of the individual amino acids, and also some kind of the small kind of the dipeptide, and also what we call the tripeptide. Okay, so now what are we going to be getting from carboxypeptidase and elastases? We are going to be getting what we call individual amino acids. We shall also have what we call the dipeptide, and also we shall also have what we call the tripeptide. Okay. Okay, such a beautiful thing. So now we have seen that these enzymes, they are going to be leading to the formation of dipeptide, tripeptide, and also some little of what we call the individual amino acids. Okay, such a beautiful thing. So now when we come over here, we have seen that we are going to be having a lot of different molecules. We are having what we call amino acids. We are also having what we call the dipeptide, and also we're also going to be having what we call the tripeptide. These are the three kind of the molecules that we are going to be having now. The question is, how are we going to be absorbing all these things into the enterocyte? Now, this one is going to be now our enterocyte. I want to first cut off this kind of the enterocyte. We try to zoom in on here and see exactly how it is going to be arranged histologically. Now, when you see these kind of the finger-like projections, these ones are what we call microvilli. Okay, so now we have said that the whole of this structure is what we call the enterocyte. Super very important. So now these kind of red kind of the groups here, the green, the blue, the black, these ones are brush border enzymes. Super very important. These are the enzymes you are going to be seeing here. This one and this one, they are the brush border enzymes. They are super very important. This enzyme here is what we call, part of the brush border enzyme is what we call the dipeptidase enzyme. Okay, is what we call the dipeptidase enzyme. Super very important. And then this enzyme here is what we call the amino peptidases. Okay, this one here is what we call the amino peptidases. Super very important in the breakdown of what we call different kind of the molecules, as you are going to be seeing. Now, the question is, these kind of the dipeptidases, which kind of these molecules are they going to be running for? At the back of your mind, how to be beautiful, they are going to be breaking what we call dipeptides. Okay, so now what happens is that dipeptides will run over on this kind of the enzyme, and then they enter through this enzyme, and then finally this enzyme will break that kind of the peptide bond, leading into the formation of individual amino acid. Okay, such a beautiful thing. So now that is for the case of dipeptide. Now, when we come over on what we call the amino peptidase, this enzyme is super very important. What happens is that this enzyme is going to be cutting off different kind of the bonds, which are going to be on what we call the amino end, or what we call the N-terminus. You know, when we call over the N-terminus, we have this long chain of what we call the amino acids. We always have what we call the carboxyl end, and the carboxyl end is what we call the C-terminus. Okay, the C-terminus. And when we talk about the amino end, is what we call the N-terminus. Okay, the N-terminus. So now what happens is that the amino peptidase enzyme, he likes to target those kind of the bonds which are close on what we call the amino end. Such a beautiful thing. So now what happens is that this kind of for example, the tripeptide, it is going to come there, and then it's also going to be coming there like this. So now this one is going to be what we call the N-terminus, and this side will be the C-terminus. What happens is that this amino peptidase will start cutting off the bond like that, and then it also comes like that. By doing that, it is also releasing different amino acids which are going to be individual. Okay, such a beautiful thing. So now we have seen that we have formed the amino acids by the influence of this kind of the enzymes you're calling the dipeptidase, and also what we call the amino peptidase. So now what is very beautiful is that we cannot break all these things into their final absorptive forms. We still have a lot and a lot of the tripeptide, and we also still have a lot and a lot of what we call the dipeptides. Okay, let us go ahead and see which kind of the mechanisms are we going to be using to absorb the dipeptide, the tripeptide, and then the individual amino acids. Okay. Now, on every cell on the human body, we have this channel, this pump, which we call sodium potassium ethipase pump. What happens is that this pump is always very serious, pumping out 3 sodium ions out, and then it is bringing in 2 potassium ions on every cell. What is this going to be the pump going to be doing? It is going to be reducing the levels of sodium inside this kind of the enterocyte. So now what happens is that we are going to be having now a reduction in what we call sodium ions within the enterocyte. Now, within the lumen, you're going to be having the foods containing a lot of the minerals, and one of them is what we call the sodium. Now, because we are having now a deficiency in what we call sodium within the enterocyte, sodium would want to move to diffuse down the concentration gradient. Okay. Now, what happens is that sodium would want to move down the concentration gradient. But as this sodium is moving down the concentration gradient, sodium is very beautiful. Like the way it did in carbohydrates, he does the same here by trying to pull the amino acids to go with. Okay, so now what happens is that he likes pulling the individual amino acids to go with it inside the enterocyte. So now what happens is that it has helped what we call the amino acids to enter into what we call the enterocyte. So now we have seen that we have entered what we call the amino acids inside what we call the enterocyte. It's such a beautiful thing. So now this channel here is also what we call the sodium glucose co-transporter, exactly the same as what we call for the case of the carbohydrates. Such a beautiful thing. So now, how are we going to be absorbing this dipeptide and also what we call the tripeptide? We have this channel on the apical or the luminal side of this enterocyte. This pump is going to be now pushing in a lot of what we call the protons into the lumen in exchange with what we call sodium ions. So it's like going to be proton sodium antiporter. Now what happens is that this pump is reducing the levels of the protons within this kind of the enterocyte, and is concentrating a lot of the hydrogen ions within the lumen. What happens that to maintain the concentration gradient, these protons, they would want to come back inside this kind of the enterocyte to make what we call the concentration to balance. What happens is that we have specific type of the channel that are going to be helping in that. We're going to be having now some of these protons which are going to be now entering through this channel, and then we shall also have some of these protons which are also going to be using this channel to enter back into what we call the enterocyte. Now, these protons are super very important. You know what they are going to be doing? Some of these protons, they are going to be helping this kind of the dipeptide to enter into the enterocyte. Such a beautiful thing. So now we are going to be seeing that some of the dipeptides are also going to be coming and they go along with this kind of the protons here. So now we have finally pushed our dipeptide inside what we call the enterocyte. Again, at the same time, we also have this channel which is pushing the protons inside. It will also help this kind of the dipeptide to enter into what we call the enterocyte. So now we have pushed again the tripeptide inside what we call the enterocyte. Okay. So now we have seen that now we have what we call dipeptide, we have what we call tripeptide, and we also have what we call the amino acids. Okay. Such a beautiful thing. So now we want to see, can we allow now the movement of the tripeptide and the tripeptide to enter into the blood? The answer is no. How do we break the tripeptide and the dipeptide into their individual amino acids? It's very simple. Within the cell which we are calling the enterocyte, we have this enzyme here. You see this enzyme here? This enzyme is very beautiful. This enzyme is what we call peptidase enzyme. Okay, it's what we call peptidase enzyme. What happens is that this peptidase enzyme will come over on this kind of the dipeptide, and it cuts over that peptide bond leading into what we call individual amino acid. Okay. At the same time, these peptidases will come over and this kind of with the tripeptide and it cuts over those kind of the peptide bonds, leading to the formation of individual amino acids. Okay. So now by doing that, by the influence of the peptidases enzyme, we are cleaving the dipeptide and the tripeptide into final individual amino acids. Okay. So now we have individual amino acids inside the enterocyte. However, there is another kind of a minor mechanism which is going to be by what we call endocytosis of what we call the tripeptide and then what we call dipeptide. We also have what we call endocytosis of what we call the dipeptide and also what we call the tripeptide. This is going to be one of the minor kind of uptake of this kind of the protein molecules into the enterocyte. Endocytosis of what we call the protein molecules. Okay. Now we have what we call individual amino acids. Now, after having the individual amino acid, we have on the basolateral surface of this cell, we have this kind of the transporter. It can now allow the exit of what we call the amino acids out into what we call the blood. So now within the blood here, we're going to be having a lot of the blood vessels like the superior mesenteric vein, the inferior mesenteric vein. This one will drive all these kind of the amino acids and then they fall into what we call the hepatic portal vein, of which the hepatic portal vein will drive these kind of the amino acids to enter into what we call the liver. Okay, so now in the liver we are going to be having a lot of what we call the amino acids. Okay, so now when we have now the proteins, the amino acids within the liver, now it is the work of the liver to start now the process of what we call the protein synthesis by joining different kind of the amino acids to make now a long chain of what we call amino acid, which we call a polypeptide, which is going to be a protein. Okay, it's all that simple. However, that process of protein synthesis within the liver is going to be under the influence of a hormone which we call the insulin, because in the process you're also going to be having release of insulin, which is also going to be going to the liver to catalyze that process of what we call the protein synthesis. Okay, it is such a beautiful thing. So now that briefly covers the protein chemical digestion and absorption in the simplest way possible by True Monad as always. Thank you very much for watching. Until we meet next time.